Today we see development in dicot embryo. Okay, this is zygote. It goes under mitotic division and it divided into two cells. This is chalizer end and this is micropyle end. Okay, the cell towards chalizer is known as terminal cell, and the cell towards micropyle is known as basal cell. Okay, then again mitotic division takes place. The terminal cell again divided into 2 and then 4. This is 4 cell terminal cells and it is known as proembryo. It is also known as proembryonal quadrate, that is 4 cells. Then basal cells also divided. Then this first cell is known as hypophysis. And this suspensor. And this is basal cell. Basal cell or last cell of basal cell is known as haustorial cells. Haustorial cells. Okay, that means from terminal cell this proembryonal quadrate is formed, and from basal cell these small cells transverse division takes place, and these cells are known as suspensor cells. Out of that, the first cell which is attached to that proembryonal embryo is known as hypophysis, and end cell is known as haustorial cell. Okay, then again mitotic division takes place, and this proembryonal quadrate is converted into proembryonal octet. That is 8 cells are formed, and this first cell is become a larger, which is known as hypophysis. Then this is suspensor and this is haustorial cell. Okay, then mitotic division takes place, but periclinal, that means only outsider cells are formed. That is, it is also known as periclinal division or periclinal mitotic division of proembryonal cells octet. And these outer cells, okay, this division takes place, and these cells are one cell is divided into 2 cells, same. Okay, this outer cells is known as dermatogen cell. Okay, dermatogen cells. Then this hypophysis also get divided into 8 cells. These are 8 cells. Okay, this is hypophysis. Then this is suspensor and this is haustorial. Now from these cells, the 4 cells towards suspensor cells, these 4 cells gives rise to a root cap and a root tip. Okay, then the 4 cells, that is these 4 cells towards proembryonal cell gives rise into root cortex. Okay, then these also get divided into 2 parts. This half part towards micropyle end. Okay, this is micropyle end, this is chalizer end. These cells towards micropyle end gives rise into hypocotyle, and the cells towards chaliza gives rise into plumule and cotyledon. Okay, these are then these suspensor cells and haustorial cells. Then these haustorial and suspensor are degenerated. Then this embryonal this whole structure is known as globular embryo. Okay, this globular embryo change their shape. Okay, and it become heart shape. Okay, this is heart shape, and this is plumule. Okay, this is root tip. Okay, this is also known as radial cells, and this is plumule and radial. From radial, root tip is arised. Okay, and this shape is known as, or this structure is known as, heart shape embryo. Further development takes place. This is gives rise, and the shape is become a horse shoe shape embryo. Okay, and at last this embryo is like this. These two are cotyledon. Okay, this central is cotyledon or cotyledonary node. Okay, the space or this terminal end is plumule, and this end is a radical. That is, from radical, root tip is arise. The space between plumule and cotyledonary axis is known as epicotyle, and the space between cotyledonary node and radical is known as hypocotyle. Okay, this is general dicot embryo. Okay, two cotyledons are present, that's why it is known as dicot. Okay, then we see development in monocot. Okay, development in monocot embryo. In monocot embryo, this zygote undergo mitotic division, and here 3 cells are formed. Okay, 1, 2 and 3. Okay, then the cells towards, this is chaliza end and this is micropyle end. This upper side cell or towards chaliza, this cell is known as terminal cell. The cell towards micropyle is known as basal cell, and this is middle cell. Okay, then this, from these middle cells, all structure are arise. That means plumule, radical, epicotyle, hypocotyle and suspensor are give rise from this middle cell. Then from terminal cell, cotyledon is arised. Okay, that is from terminal cell, this cotyledon is arised. Okay, and this from middle cell, all structures, that is plumule, radical, epicotyle and all these structures are arised. And from this basal cell, it remain as it is. Okay, then again division is takes place, and this structure is formed, that is this monocot embryo. Okay, this end or these cells are known as plumule, but in monocot it is known as coleoptile. That is from coleoptile, that is plumule in dicot, and this structure is known as coleorhiza, that is radical, but in monocot it is known as coleorhiza. This is basal cell. Then from these structures are arised from middle cell. This is suspensor. Then this one is cotyledonary axis Okay, and from this structure is cotyledon. Single cotyledon is present, that means monocot. Okay, later these suspensor and basal cells are degenerated. Okay, and the structure is look like this. This upper side is coleoptile. This is coleorhiza, and this is cotyledon. Okay, in the monocot, that means in maize the structure look like this. Okay, this is maize seed. Okay, this is divided into two parts. Lower part, this embryo is present. Okay, this is cotyledon, this is coleoptile, this is coleorhiza. Okay, and in upper side it is again divided into two parts. Outer side, here storage reserve food material is present in the form of protein. It is also known as alluron. Okay, and the reserve food material in the form of starch is present in the center. Okay, then reserve food material for this developmental embryo is coming from this upper sides, not from cotyledon. These proteins are come into cotyledon, and from cotyledon it is supplies to developmental embryo. Okay, this is development in embryo. Okay. In next part we will see about seed and fruit.